Hey Biotherians, and welcome to our second discussion in the population genetics unit. Today I'm going to be discussing the Hardy-Weinberg Principle. Now, last lesson or last video, I discussed the three types of frequencies that you can use to determine the characteristics of a gene pool of a population. You can have either genotype frequencies, which indicates the frequency of either the homozygous dominant genotype, the heterozygous genotype configuration, or the homozygous recessive configuration of a pair of alleles. You can also have what's called the phenotype ratio or the phenotype frequency, which indicates the two different, if it's a dominant versus recessive trait, the two different phenotypes that are basically going to show up in a population. So, for example, I think the example I used in the last video was the fur of mice, where you had black being the dominant phenotype and white being the recessive phenotype. This is obviously the easiest frequency to calculate, because all you have to do is count the phenotypes in the population. That's the easiest one to basically determine. You can also have what's called the allelic frequency, which is basically the frequency of either the dominant or the recessive allele in that population. Now, this all sort of plays together when you're dealing with this Hardy-Weinberg principle, which basically states that if the allelic frequencies of a population will remain constant if five things are happening. So basically, a population's allelic frequency, so let's say you had a frequency of 60% of the dominant allele and 40% of the recessive allele, as I indicated in our last video in the example there, that ratio is going to stay the same if five things happen in the population. Now, this rarely ever happens in nature, but if these all occur in nature, then in theory, an allelic frequency will not change in a population. Now, what are the five things? Well, the five things are as follows. Number one, the population has to be extremely large. You got to be dealing with a very large population that can basically be so large that if any random chance events occur, maybe a disaster or something like that, it's not going to affect the overall allelic frequency. Now, I will discuss later in the unit how these allelic frequencies can change when a disaster or a kind of a random occurrence occurs, but I'll get to that later in the unit. But for now, be aware, for Hardy-Weinberg, you've got to be dealing with a very large population of organisms. Secondly, the mates have to be chosen randomly. Again, that doesn't happen very often in nature. Usually there is some kind of selective breeding that's going on. Females are selectively choosing their males. But if there is random mating in the population, then the allelic frequencies will also stay the same. There's also no net mutation. So it's basically indicating that, there's going to be no beneficial or harmful mutations that are going to basically cause a shift in the frequency of the alleles based on a mutation that occurs in that particular gene that controls a specific trait. There can be no migration. The organisms that are in that population must be staying put. They can't be moving around. Again, not very likely, but there are populations in the wild that aren't able to migrate. For example, there could be an island population of organisms. You could be talking about like populations in isolated areas like the Ngoro crater in Africa. You've got populations of lions there that are basically isolated inside the crater. They can't get in, and those lions can't get out, or there are no other lions from other populations can get in. So there are instances where this can occur, and there's no natural selection occurring whatsoever. So these are all five of these have to occur in order for an allelic frequency in a population to remain constant. Again, it doesn't happen very often. And again, this is why natural selection is occurring, because some of these things are happening. There are going to be shifts in natural populations with allelic frequencies. But these two gentlemen in 1908, Hardy and Weinberg, came up with a mathematical equation to basically calculate if I know, for example, an allelic frequency, or if I know a genotype frequency, or if I know a phenotype frequency, I can use that information to calculate other frequencies that aren't as maybe readily available. For example, the most likely phenotype, the most likely frequency that you would know as a biologist if you're going out to count a population of organisms, would be the phenotype frequency. Now, from that information, if your population is in Hardy-Weinberg equilibrium, which means that it has all those 5 characteristics going on, you can use a mathematical equation to calculate from those phenotype frequencies. You can actually deduce what the genotype or what the allelic, actually both, you can deduce both the genotype and the allelic frequencies of that population. Now let me explain how that works. At the base of the equation is this. The base equation for Hardy-Weinberg is a very simple one. It's P plus Q equals 1. Now, first of all, this equation is somewhat given to you in your data sheet. There are actually 2 Hardy-Weinberg equations, which I'll be getting to both today. The one that you're given is the one at the bottom, the expanded equation. I'll get to that one in a couple of seconds. This equation up here, P plus Q equals 1, is not in your data sheets. This is one that you need to be able to remember on your own. The government gives you this information. They don't give you this information. So, but if you can remember this one on your own, that's going to make your life considerably easier, as I'll explain here in a couple of seconds. What do the P and the Q represent? Well, the P plus Q equals 1 equation is basically going to allow you to calculate, the only frequency this will allow you to calculate are allelic frequencies. So P plus Q represent the dominant allele, P, that would be the dominant allelic frequency, and the recessive allelic frequency, which would be Q. So P equals the dominant allelic frequency, Q represents the recessive allelic frequency. So for example, I mentioned earlier in today's lesson and in the last lesson with the mice, the allelic frequency I think I gave you was 0.6 and 0.4, right? So you had 60% of the alleles in the population were dominant alleles, and 40% of the alleles in that population of mice were recessive alleles. So 0.6 plus 0.4 equals, well, guess what? That equals 1. Again, 1 equals 100%. So this is basically saying that the percentage of dominant alleles plus the percentage of recessive alleles should equal 100%, or if I have them as decimals, 0.6 plus 0.4 would equal 1. It's a pretty standard, straightforward equation. But, and that's the other thing you got to remember about these equations. They always have to equal 1. If you get numbers that add up to more than 100, or more than 1, let's say you get an answer that equals, let's say you calculate all your frequencies and you get a number of 1.05, or you get a number of 0.95, that means that population is not exactly in Hardy-Weinberg equilibrium. If all the frequencies add up to 1, then you know that, okay, this population is actually in Hardy-Weinberg equilibrium. It's got the 5 characteristics that are necessary: large population, mates are chosen at random, no net mutations, no migration, no natural selection. So in order to calculate whether a population is in Hardy-Weinberg equilibrium, you just basically add up your frequencies once you've calculated them. And if they don't add up to 1, you know that your population isn't exactly in Hardy-Weinberg equilibrium. All right? But the starting equation is P plus Q equals 1. That's the equation you will always use to calculate allelic frequencies. You can't use this equation directly to calculate your geno and your phenotype frequencies, but you can indirectly use this equation, as I will get to here in a couple of minutes. Now, if you're a math head, and I know some of you in biology are actually math heads, you could expand this equation out. If you took 1 squared, which is essentially going to still be 1, if I squared my 1 and I also square my P plus Q, if I square P plus Q in math, I'm going to get this equation. P squared plus 2PQ plus Q squared now still equals 1. This equation, the one at the bottom, the expanded Hardy-Weinberg equation, which is the one they give you in your data booklet, this equation you can use to calculate both your geno and your phenotype frequencies. Let me explain. P squared in the equation is going to equal the frequency of organisms that have a homozygous dominant genotype. So in the last video I discussed, it was, I believe the letter I used was B for coloration of the fur. The capital B was black, little b was white. So in the case of the last day's video, that would be your capital B genotype. homozygous dominant. The 2PQ part of the equation is going to represent your heterozygous organisms, their frequency. So how many heterozygous organisms, or what's the frequency of heterozygotes in the population? This would be, in the equation I gave last day, it would be the capital B, little b variation of the genes, right? That would be the heterozygous combination. So you could use this part of the equation to calculate the heterozygous organisms in their frequency in the population. The Q squared portion of the equation is going to equal the homozygous recessive organisms in the population. So these would be the little b's based on the example from last day's video. So just to recap this expanded equation, the top equation allows you to calculate your allelic frequencies. The bottom equation allows you to calculate your genotype frequencies. So you've got either organisms that are homozygous dominant, P squared, organisms that are heterozygous, 2PQ, or you've got your organisms that are homozygous recessive, Q squared. Again, you're going to have three frequencies that in theory, if you add them all together, you should get a total frequency equal to 1, right? 100%, right? The three groups of organisms are going to equal that 100% total. If the population is in Hardy-Weinberg equilibrium, they should all equal out to 1, or the equivalent of 100%. All right? Now, this equation can also be used to calculate your phenotype frequencies, because you understand that the organisms that are homozygous dominant and the organisms that are heterozygous are both going to be showing the dominant phenotype. So if I wanted to calculate the organisms in a population that had the dominant phenotype frequency, or what the frequency of the dominant phenotype is, pardon me, I would calculate it based on P squared plus 2PQ. So this part of the equation here, the P squared plus 2PQ, if I add those together, that in theory should equal the amount of organisms that have the dominant phenotype, not the amount, the frequency of organisms, the percentage of organisms in the population that have the dominant phenotype. The good news is Q squared, pardon me. Q squared is my homozygous recessive organisms. Well, I know that only homozygous recessive organisms are going to have the recessive phenotype. So the nice thing is Q squared, which equals your homozygous recessive genotype frequency, also is able to give us our recessive phenotype frequency as well. So this equation at the bottom can be used to calculate both genotype as well as phenotype frequencies. The P plus Q equals 1 equation is only going to be used for allelic frequencies, and this is the equation that you don't get. You've got to remember this one and why you would need that equation. P plus Q equals 1 is going to be my allelic frequencies. All right? A lot of information about it in the notes as well if you've been following along with the notes. Hopefully you've got your notes open today, because I'm going to give some examples directly out of the notes. They're not going to be up on the whiteboard, so if you want to pull those notes up right now before I get started, maybe get a calculator as well. You can try some of these questions at home while you're watching the video. Maybe block out the bottom of the slide so you don't see the numbers, so you can actually maybe try some of these equations on your own. I'm going to give you some time to work on them. This little graph here, you're not ever going to really see this, but there is a bit of a trend. If you begin to run these equations, you'll begin to see that there is a distinctive pattern. For example, if I've got a population, if I've got a population that has 50% heterozygotes in it, if I'm following Hardy-Weinberg equilibrium rules, then in theory, 25% of those organisms are going to be homozygous dominant, and 25% of those are going to be homozygous recessive. There is a bit of a trend that you can actually begin to mathematically see. If you're good with numbers and good with math, you'll begin to see relative patterns in it. You would never be asked about this on an exam, but it is kind of a nice chart to show you the relationship that as, for example, my homozygous recessives drop in number, what is happening to my homozygous dominance, what's happening to my heterozygotes at the same time. There is very much a pattern that plays out because it is a mathematical relationship. Let's take a look at an example. Now, again, if you flip the page, don't cover the bottom of the page. My page here is not showing anything. yours will automatically show up if you've got the PDF files at home. Let's take a look at an example and see if we can do some Hardy-Weinberg work on this one. I got a population that has two alleles. I got a capital R and a little r for a particular gene. I don't really need to know what the gene does, but we've got two alleles, capital R, little r, dominant versus recessive. The allelic frequency of capital R is 20%. What I want you to do is I want you to calculate, based on knowing what the allelic frequency of R is, capital R, I want you to calculate the frequencies of the homozygous dominant organisms, the heterozygotes in the population, and the homozygous recessives. So you're going to need to use both the equations here to calculate what you need to calculate. Right now, I'll give you a clue. Capital R represents P. That's the P value of the equation, and we're going to have to convert that 20% down to a decimal. So I'm going to start you off with a little bit of a clue on this one. P is going to be equal to 0.2. That's our starting point. Now you need to calculate based on P equaling 0.2, can you calculate P squared, 2PQ, and Q squared? Give it a shot. Pause me if you need to. I'm going to go over the information right away here, starting right now. Hopefully you tried it out on your own. As I mentioned to start off with, P is going to represent the frequency of the dominant allele, which is capital R. I just mentioned that before you paused me. If you did pause me, that's a value of 0.2. Well, if I know my value of P is 0.2, and if my population is in Hardy-Weinberg equilibrium, I can plug 0.2 into my shortened equation to calculate Q. If I know that P is 0.2, well, then if I plug it into my equation on the top, Q is going to give me a value of 0.8. So I'm going to have 80% of the alleles in the population are going to be recessive alleles. So I've got a P value now of 0.2. I got a Q value of 0.8. Now I can use my P and Q values to calculate my homozygous dominance, my heterozygotes, and my homozygous recessives using the expanded equation. So to get my homozygous dominance, I'm going to take a 0.2 and I'm going to square that. That's going to give me a value of, well, I've got the whole thing expanded here. I'm going to just bring it down here. If I square 0.2 squared is going to give me a value of 0.04. So technically in this population, 4% of the organisms are going to be homozygous dominant. If I plug in 2PQ, I'm going to plug in 2 times 0.2 times 0.8. That's going to give me a value of 0.32. So 32% of the organisms, 32% of the organisms in this population are going to be heterozygotes. And then if I plug in my Q value, which is 0.8, I'm going to square that. I'm going to get a value of 0.64. That means that 64% of the population is going to have the homozygous recessive genotype configuration. So there you go. From that information, I can also calculate which of the organisms, what the frequencies are for the phenotypes. That's not part of this question, but I could quickly do that very quickly as well. If I took my 0.4 value and my 0.32 value, that's going to be these two values here. That's going to represent, if I add those two together, that's going to be the frequency of my dominant phenotype. So my dominant phenotype frequency for this population is going to be 0.36. My recessive frequency, easy to say, recessive phenotype frequency is going to be 0.64. It's going to still be equal to Q squared. That value doesn't change. I know that wasn't part of the equation or the question, but I just wanted to show you how you could quickly expand out to calculate your phenotype frequencies now that you know your 3 genotype frequencies. It's a lot easier to calculate your phenotype frequencies from your genotype frequencies. Very difficult if you're given the dominant phenotype frequency, that doesn't help you. You need to know if you're given phenotype frequencies, the one that you need to isolate is going to be the recessive phenotype frequency, because if you know that, you know that that's a value of Q squared, and I can take that frequency and I can get the square root of it. That gives me a Q value, and then for my Q value, I can then calculate my P value. So generally speaking, with these equations, what you want to do is you have to isolate P and Q as quickly as possible. If you can isolate and get the values for P and Q, then as soon as you understand what the P and the Q values are, then it's just a matter of plugging them into the expanded equation to get either your geno or your phenotype frequencies that the question is asking. Let's go over another one. Losing my voice here. It's got to be the end of the semester almost, isn't it? All right, so we can modify the equation. We can also calculate population sizes. So if I know the number of organisms in a population, for example, I can calculate with the frequencies. I can then calculate in a population. So let's say I had a population of 200. Let's use the example from the previous page. I've got a population of 200. Well, how would I calculate that? Give you a second if you want to just do the calculations. Once you know the frequencies, and all you have to do is take the number of organisms in the population and times it by the frequency, right? So I know that my frequency of homozygous dominance, for example, was 0.04. If I times that by 200, that means that I'm going to have 8 organisms in that population of 200 that are going to be homozygous dominant. I have 0.32 as my heterozygous genotype frequency, times that by 200. That's going to give me 64 organisms in that population of 200 that are going to have the heterozygous configuration. And I had a 0.64 homozygous recessive genotype frequency. If I times that by 200, that's going to give you 128 animals or organisms in that population that are going to have the homozygous recessive configuration. So once you know the frequencies, you can then use that information if you're given the number of a population. It's just calculating, if I've got 1,000 organisms, well, if I know the percentages, I'm just going to calculate the percentages based off of 1,000 organisms. So that's not that hard to kind of extrapolate. But again, those could be questions you could see coming up on worksheets, on quizzes, on exams, on the diploma. Those are legitimate questions that could come up with Hardy-Weinberg questions. Now, as I mentioned earlier, Hardy-Weinberg rarely occurs in natural populations. To meet all 5 of those conditions that I talked about, it's very difficult to meet even 2 or 3 of them, let alone all 5 of them. But this calculational, this formula, this principle allows us to measure the amount of variation within a gene pool. If I run the numbers in, let's say, 2010, go back to a population 5 years later, I can check based on, I can check the number in the population, what the phenotypes are, and then I can run numbers to calculate, is there a shift in the gene pool? Have the numbers significantly changed between 2010 and 2015? And if so, then is there some kind of a microevolution that's going on in that population? So even though a lot of populations don't qualify to use Hardy-Weinberg, we can still use this formula to have, for biologists can use it to sort of calculate trends in a population. Is there a shift toward one allele or the other? That's where this equation can come in very handy if you're studying populations for long periods of time. Again, if there is no allelic frequency in a population over a long period of time, that's given the name a population that is in what we call genetic equilibrium. If that population is just happy as, a pig and poop, as it were, and there's nothing that's driving that population to change its allelic frequencies, it's going to basically be what's been called genetic equilibrium. And again, as I mentioned, gradual changes in allelic frequencies are going to be considered to be microevolution, which could be a precursor to perhaps, if it continues long enough, could we see some form of speciation occurring where one population maybe evolves into another species altogether. That can occur if microevolution is allowed to continue for long periods of time. But as you might remember in Bio 20, in order for evolution to occur, for natural selection to occur, you can't have migration, right? You can't have organisms entering and exiting the gene pool because that will pollute the gene pool with new DNA that's coming in from outside. You have to isolate a population in order for natural selection to occur. So you can see why that needs to happen. No migration can occur if you don't want the allelic frequencies to change. If you, there is that connection between this and natural selection. That's it. That's it for today. I got one more topic related to the concept of allelic frequencies and gene pool and Hardy-Weinberg. Next day we're going to be discussing here's the reasons why a gene pool wouldn't change, but how it happens to actually cause gene pools to change. That'll be my final discussion before you get your essentially your first homework assignment in this unit. We'll be discussing that, causes of gene pool change. Thanks for tuning in, everybody, and we will see you again very shortly. Take care.